This conference will now be recorded.
Hello and welcome back on my continuing journey with Grand Canyon University. My name is Viv and today we're going to talk about fluid compartments of the body. We're looking microscopically at where the body holds fluid and what kind of materials are floating around in it. This video is made for students in higher education who should have a background in chemistry and anatomy and physiology. There are two major compartments which hold fluid in the body. I'd like to take a moment to preface that when I say fluid, I am talking about water in the body that has ions in it. I may use the terms water and fluid interchangeably, but I am talking about the nutrient-rich water. Intracellular fluid, or ICF, is fluid that is within the membrane of individual cells, and we have trillions of cells in our body. Extracellular fluid, or ECF, is fluid found outside of the cell. Considering mostly blood plasma and interstitial fluid, the fluid between the capillaries and cell. We'll take a closer look at those compartments and we'll discuss the different ionic makeups of each. We'll end by looking at a recent study that asks the question, how long is Zika detectable in our body fluid? There are two fluid compartments in the body, and we are not talking macroscopically, like the stomach or the chambers of the heart. We're talking about microscopic, where you would need an electron microscope to hone in on where the fluids are being held. These are intracellular and extracellular fluid. Intracellular fluid makes up 66% of our total fluid volume and is found within the cells. Remember that our cells have a fatty bilipid plasma membrane that does not allow for free flow of fluids and nutrients to come and go as they please. Our cells have specific channels and processes that allow for the exchange and release of materials. Within the cell, of course, there is water, but there is other materials that make up the ICF, which is a little bit of sodium, a lot of potassium, and some other cations, a lot of phosphate, and a little of other proteins. This makeup of ions in the ICF is different from the concentrations in the ECF. Our extracellular fluid is made up of blood plasma and interstitial fluid and makes up about 33% of our total fluid volume. These two compartments can freely move ions between each other, but plasma does contain bigger proteins and molecules that have specific entry and exit points. The compartments contain a lot of sodium and a little potassium, a decent amount of bicarbonate, a lot of chlorine, and a little of the other stuff, especially in the plasma. Plasma only makes up about 20% of the ECF, where the interstitial fluid makes up about the other 80%. Again, it just goes to show how massive of a multicellular organism humans are, that our blood plasma is 1/4 the volume of interstitial fluid, and that within our cells, it contains 2/3 of our total volume. That's wild. So let's take another look at the ionic makeups of these areas. In green, we have our cells, which hosts the ICF. The main cation is potassium and the main anion is phosphate. The separation of fluids outside and inside the cell maintains cell homeostasis through the fluid. The separation of fluids outside and inside the cell maintains cell homeostasis through the plasma membrane. In light blue, we have our interstitial fluid, the fluid between the blood capillaries and the cells, whose main cation is sodium and the main anion is chlorine. Finally, we have the blood plasma in red, who also has a majority sodium and chlorine. This is because capillaries and interstitial fluid have the ability to easily move materials between each other using the pores on blood vessel walls. The capillaries, and thus blood plasma, will host bigger molecules like proteins, red blood cells, glucose, hormones, and other building block materials that the interstitial fluid will not contain. It is important to note that when changes happen to the plasma, it is reflected in the interstitial fluid since the permeability between the two of them is so free. Two other minuscule compartments of extracellular fluid are the lymphatic and the transcellular fluids. The lymphatic system takes interstitial fluid and filters it through the lymph nodes for immune defense purposes and adds it back to the plasma. Additionally, there are a bunch of transcellular fluid volumes in different body cavities, including cerebrospinal fluid found surrounding the brain and spinal cord, intraocular fluid that shapes and nourishes the eyes, synovial fluid, which lubricates the joints, pericardia, intrapleural, and peritoneal fluids, which surround the hearts, lungs, and intestines, respectively, and finally, the digestive juices and digestive foods within the stomach. The purpose of this fluid and the ionic balance is important in maintaining homeostasis. The extracellular fluid balance is controlled by volume and osmolarity, which is measured in milliosmoles of solute over liters of solution. ECF controls regulation of blood pressure and salt balance. When plasma is reduced, materials from the interstitial fluid move in to take its place to make up a part of the loss. Blood volume is needed to maintain pressure in the arteries for the heart to beat properly. Additionally, ECF maintains the salt balance of the body. Salt, or sodium chloride, is an electrolyte, which means that it breaks down into its individual atoms when mixed with water, which is why we see such high amounts of sodium and chlorine in the extracellular fluid makeup. We cannot make salt ourselves, which is why herbivorous animals will travel to salt licks and why we add salt to foods. Salt filtration is done in the kidneys, where the amount of salt we ingest is the same amount we excrete. Salt and water follow each other around to maintain osmolarity. A recent observational study coming out of the United States wanted to collect information on Zika virus infection and diagnostics to be able to protect pregnant women and travelers. The information collected would give an idea about how long the virus lasts in the different fluids of the body. Zika is a virus that is usually asymptomatic, but can produce symptoms like fever, rash, muscle pain, joint pain, or headaches. In most cases, people recover and it has a low mortality rate. Zika is dangerous for pregnant women, though, because it can cause birth defects on the fetus, including microcephaly, where the brain is underdeveloped, or Guillain-Barré syndrome, which is a sickness of the nervous system. To diagnose someone with Zika, they must have detectable amounts of the Zika virus RNA or specific antibodies. This study aimed to find out how long these detectable levels last in the fluids of the body. Donors were chosen after donating blood at specific collection sites who were confirmed to have the Zika infection. 53 asymptomatic donors were chosen to participate in the experiment after nucleic acid amplification test, or NAAT, were ran on the plasma samples and then confirmed with polymerase chain reaction, or PCR, assays. Researchers collected 75 milliliters of anticoagulated blood, 7 milliliters of blood in a specialized tube for gene expression analysis, 15 milliliters of urine, 3 milliliters of saliva, 1 to 5 milliliters of semen, because again, we're concerned about pregnancies, and cheek and nose swabs. This collection of material was ran for Zika virus RNA amounts using PCR and NAAT. Over the span of a year, donors provided samples at least 6 times at weeks 1, 3, 6, and months 3, 6, 9, and 12. And yes, that is technically 7 visits, but only 75% of the participants came at least 6 times. The endpoint goal of this experiment is to no longer detect Zika virus RNA in the patients. At the beginning of the experiment, all the patients were confirmed to have asymptomatic Zika with detectable plasma RNA. Most of the patients developed antibodies for the Zika virus before the first follow-up, making the mean time for detectable infection to developing Zika immune cells around 7.5 days. This experiment determined that most participants had red blood cell associated Zika virus RNA infection in early infection and up to 3 months after initial infection, where 75% of the participants had this Zika RNA detected in their whole blood. They determined as well that prepackaged blood hosts the Zika RNA longer since there is nothing to dilute the infection in the red blood cells. Interestingly, though, 25% of the participants did not have any red blood cell associated Zika RNA, which the authors noted should be investigated further. Additionally, 43% of patients had Zika RNA detected in their urine with mean duration between 14 and 23 days, and 35% of participants had detectable RNA in their saliva for a mean time around 26 and a half days. The authors note that those who had dengue virus, which is a virus that produces flu-like symptoms before Zika, cleared from the saliva quicker than those who had never had dengue. Finally, of the 12 miles providing semen samples, only 25% had detectable Zika RNA. The research concludes that whole blood testing is the best diagnostic tool for the Zika virus. This experiment not only validates the testing procedures, but lays the foundation for vaccine development and therapy. There is so much more to this experiment that I couldn't talk about, like the dengue fever factor, when participants developed their antibodies, what kind of antibodies they developed, etc. But I wanted to focus on the fluids as much as possible. So, our bodies have 2 compartments which hold 42 liters of bodily fluid. 28 liters are within the cell, or intracellular fluid. It is composed mostly of potassium and phosphate. The other 14 liters are composed of plasma and interstitial fluid, making up the extracellular fluid. Its main components are sodium and chloride, the ions that form salt. The ECF is a regulator of salt and blood pressure. Scientists have been studying how long the Zika virus lasts in our bodily fluids. They have found that Zika RNA maintains its longest stint around 3 months, associated with red blood cells found in the plasma. It's a good thing then that our interstitial fluid and the lipid membrane on our cells provide a barrier to this virus. I absolutely recommend reading the article, especially if you're from Puerto Rico, where a lot of the participants were from, or if you've ever had dengue fever. The ECF and ICF make up about 60% of body weight, 40% of that being intracellular. Remember to take care of your body and to please watch your salt intake. If you are traveling to a country with Zika outbreak, especially if you are a woman, make sure that you wait and have your blood tested before getting pregnant. Here are my resources. Thank you so much for watching, and I will see you the next time.